Hi everyone, this is lecture 28, Sex-Linked Traits, and this material is found in chapter 12 in your textbook, page 339 to 343. Up to this point we've talked about the chi-square test and how it's used to test the goodness of fit for a hypothesis. We've also talked about standard Mendelian traits such as using like monohybrid and dihybrid crosses. Mendel's crosses in his crosses he discovered the simple principles of Mendelian genetics, but he didn't discover the more complex genetic traits that we know of today. A lot of times when a chi-square test is performed on a genetic cross, it will fail. And it will fail because the phenotypes don't match the 3-to-1 ratio or the 9-to-3-to-1 ratio. And so over the next couple of lectures, we're going to be talking about some extensions of Mendel, things that he didn't discover, but that we now know are really important in genetics. Sex-linked traits, of course, are one of those. So number one, traits that occur almost entirely in males and skip generations in families are usually. Number two, red-green colorblindness in humans is a sex-linked recessive trait. If a colorblind male marries a female with normal vision, what percent of their male children will likely be colorblind? So we use the word sex-linked to refer to genes that are on one of the two sex chromosomes, in the sense they are linked to that chromosome. The X chromosome contains about 1,000 or so genes. And so most of the sex-linked traits we'll be talking about are X-linked. The phenotypes that you observe in females and males differ because males have one X and females have two. It can lead to very different phenotype outcomes. The Y chromosome has a very small number of genes, about 75. So there's not as many Y-linked traits. However, we've talked about one of them so far, the SRY gene, and how it determines maleness. We're going to talk about an X-linked trait that causes red-green color blindness. And this gene is called an opsin gene. Opsins are a family of genes, one member of which is rhodopsin, which is shown here. Rhodopsin is found in rod cells in your retina, which is shown here on the left-hand side. The rhodopsin gene produces a protein which spans your membrane, the plasma membrane in a retina cell, and senses light. When people have mutations in rhodopsin, it can lead to night blindness because they don't see light well in dim light. Rhodopsin is one of a family of genes. There are other opsin genes that detect color light. We have 3 of them in humans and primates. We have the red opsin gene, the green, and the blue. Each one is expressed in a different specialized cell in your retina. They're called cone cells. The red cone cells express the red opsin. The green cone cells express the green opsin. And the blue cone cells express the blue opsin. The blue opsin gene is found on an autosome, whereas the red and the green are found on the X chromosome in humans. We say that we have trichromatic vision because we have 3 genes that detect 3 different colors. This diagram shows the spectrum of light that these different opsin genes detect. The blue or short wavelength opsin 2 gene, the medium or green opsin 1 gene, and the long or red and opsin 3 gene. We're going to talk primarily about the opsin 3 gene. So how does red-green color blindness happen? Well, it's due to chromosomal mutations. Usually there is chromosome X rearrangements, for example inversions or duplications or deletions that involve the red-green opsins. These genes are disrupted and the proteins aren't made properly. Again, the gene is called opsin 3 for the red or long wavelength opsin, and the symbol is OPN3. Now, for X-linked traits, we needed to note that the gene is on the X chromosome. So in order to do that, when we write the genotypes, we always use the X chromosome and superscript gene symbol. For example, in this diagram, this table, X superscript OPN3. Because the normal vision opsin is dominant, I'm using capital letters, OPN3 uppercase letters. A female who is homozygous for 2 normal copies, the genotype would be rewritten like this. She would be homozygous, and she would have normal vision. If a female is a carrier for 1 mutation for an allele that causes red-green color blindness, her genotype would be written this way. Uppercase OPN3, lowercase, dominant recessive. She's a carrier, but she has normal vision. Again, the normal vision is dominant. However, if a female has 2 red-green colorblind alleles, lowercase OPN3, she is homozygous but will have red-green color blindness. This is very rare though in women because this allele is very infrequent in the population. The frequency of a female having both of those mutant alleles would be the product, multiplying the frequency of that allele times itself in the population. It's a very low number usually. However, many females could be carriers. For an X-linked recessive allele, in females, 2 mutant alleles are required for color blindness. Again, it's very rare. So for this reason, recessive traits tend to skip generations because they are masked by the dominant allele. Also, as we'll see in a minute, as you can see from this slide, females are not likely to have it. For males, it's a different scenario. Because we only have 1 X chromosome, the second chromosome is just denoted as a Y. Make sure that you never include an X-linked trait on a Y chromosome. It just doesn't exist. For a male who has 1 X chromosome with a normal vision allele, we use a new term. He is not homozygous or heterozygous because he only has 1. We use the word hemi to refer to half. Hemizygous, half the normal copy. For a male with this genotype, he would have normal vision. For a male with a red-green colorblind allele, though, he would also be hemizygous, but have red-green color blindness. So, for X-linked recessive alleles, males only require 1 of those mutant alleles to express that trait. And that's why it's more common in males than females. Instead of having to inherit 2, we only have to inherit 1. When fertilization happens, the probability of getting someone having a female with a trait versus a male with a trait is different, and that's what's special about the X-linked traits and Y-linked traits. For example, let's take a female who's a carrier. She has 2 alleles, one's a normal vision and one's colorblind vision. Half of her gametes will have the normal, half of her gametes will have the colorblind. Let's say that her partner is a male who has normal vision. He has an X chromosome with the normal copy, and he has a Y chromosome in half of his gametes. Remember, there are no X-linked traits on Y chromosomes. Setting up a Punnett square then is relatively simple. A quarter of the offspring would be female that are homozygous for normal vision. A quarter of the offspring would be heterozygous for normal vision. A quarter of the offspring would be men, or be boys that have normal vision, and a quarter would be boys that have red-green color blindness. When you factor into the phenotype ratios, all of the girls from this cross would have normal vision, but half of the boys would have red-green color blindness. All right, that's all for today. I will see you in class.